Okay, so in this video, I'm going to be showing you the results of our pGLO transformation experiment. And so out here on my benchtop, I've got all of our plates. And it's been just about 24 hours. And the good news is that we got some bacterial transformation, not as much as I usually get. However, these supplies were a little bit old. These were supplies that were ordered for spring semester, and they were kept in the refrigerator. And so the bacteria may not have been all that great, but still worked. Usually I get more transformed cells than we have on these plates, but we have something to look at, and so that's fantastic. So let me remind you again of what our starter plate looked like. So I kept our starter plate, and at this point I don't care about being sterile. I don't want to get anything, I don't want to ingest anything, but I don't care about the plates getting other stuff on them because this is the end of the experiment. So I'm going to go ahead and I'm going to take the top off of my Petri dish. So this was the starter plate that I took material from. Okay, so remember, there are going to be some questions for this week. That's really your assignment, is to answer questions. And in one of those questions, you're going to have to describe what the starter plate looked like. And so here's that lawn of bacteria, right? And I tried to thin it out as best I could. And I did get some individual colonies in there. Bacteria is white, a little stinky. If you were here, you could smell it. And the colonies are about a millimeter, half a millimeter in diameter. Now they've grown a little bit more. They're over 48 hours on this plate, but I still can see some individual colonies on there. Let me hold it closer to the camera, just so that you can see. You see like streaked lines, right? But if you see those little circles that are in there, there's some throughout here, and again, some throughout here. Okay. Let me show you the other starter plate. There might be some better, there's some better individual colonies on this one. Okay, so wherever you see those individual dots, those are individual colonies. And that's what I grabbed off this plate. Remember, one colony is the result of one bacteria dividing. And the E. coli divide really quick, which makes them great organisms to study transformation. And you can really tell if the trait's being passed on. Replicates relatively quickly. These E. coli that we use here in the lab are non-pathogenic, which means that they do not cause disease. They're education grade E. coli. They're not going to hurt me at all. Okay? Okay. Remember, our starter plates, we took bacteria that we rehydrated for 24 hours, and we plated them on these plates that only contained Loria broth. So LB stands for Loria broth. Remember, that is just nutrient-rich medium. Nothing special is added to it. And so bacteria, E. coli, love to grow on this stuff. They thrive on it. Okay. So then remember, we set up two microcentrifuge tubes. One we wrote plus pGLO, the other we wrote minus pGLO. Okay. We added bacteria to both tubes. And then we added pGLO plasmid DNA to just the plus pGLO tube. We did not add that DNA to the minus pGLO tube. So all of the plates that we made from the minus pGLO tube, those are our control plates. Okay? And that's the variable, that's the manipulated variable here in this experiment, is the presence or absence of the pGLO plasmid. Okay, remember the pGLO plasmid has some really cool genes on it. Specifically, it has the ampicillin resistance gene, so it can grow. Those bacteria that take up the plasmid and express it are going to be able to grow in the presence of the antibiotic ampicillin. And it also has the green fluorescent protein gene. And so those organisms that are living in the presence of the sugar arabinose are going to have that gene turned on, and therefore are going to express the green fluorescent protein. And only in the presence of ultraviolet light, UV light, that protein will glow. Okay, so lots of gene regulation happening here. So let's look at the first set of plates. So I took both the plus pGLO tube and the minus pGLO tube, oh, these stink, and I plated them onto agar plates that just had Loria broth. Okay, so again, this is just nutrient agar medium. Here's the result of my plus pGLO tube. So you can see there's a whole lawn of bacteria. Remember, I wasn't trying to thin these out at all, so that's all bacterial growth on there. We put kind of a lot of bacteria on there, 100 microliters, and then I just took my loop and I just kind of smeared it around. So all of that material is E. coli. And again, it's kind of a milky white, a little stinky, and it's a bacterial lawn, so lots of bacteria growing there. So this is the plus pGLO. So again, this is just nutrient agar, and these are bacteria that do have the plasmid. Some of them probably don't have the plasmid, so some are transformed, some are not. There's no antibiotic there, so everybody's going to grow. Okay, both the transformed as well as the non-transformed bacteria from that tube are going to be capable of growing right on this dish. Okay, here is the LB plate, so just L'Oreal broth, from the minus pGLO tube. They look real similar, right? So if I were to compare the two, I would say there's really no visible difference, right? They both grow in a bacterial lawn. They both smell, I'll tell you that. And they both have the same milky white appearance. So really no difference. The amount of growth on both is pretty much the same. Okay, so remember the plus pGLO and the minus pGLO tubes, when I plate them just on the L'Oreal broth agar, they have the same results pretty much. Really can't tell the difference between the two. And both look pretty much the same as the starter plates. Okay? Okay, the next set of plates that we're going to look at, again, still the plus and minus pGLO. These plates are going to have L'Oreal broth and the antibiotic ampicillin. And on my plate, on the bottom, right, this says plus pGLO, and then it says LB/amp. So it's L'Oreal broth with the antibiotic ampicillin added. Okay, let's look at the minus pGLO plate first. So if you remember back to the first video, which was me yesterday, I told you that the LB amp plate had some fungal growth on it. Okay, so we have to take into account that. So let's take a look. So that circle of fuzz around it, that's a fungus I got on my plate. So sometimes, as sterile as we try to be, we still sometimes get some critters on there that we don't want. But that's all that's on this plate. If you look at all the streak marks that I made, there's no little circles, no colonies. Okay, let me shine a UV light on this. And I'll do this again with the lights off in just a second. I'm not sure if you can tell. There is nothing glowing on my plate. I can see the UV light. There's nothing glowing back at me. Okay? So, if we were to compare the LB amp plate, that one, to my plus pGLO plate, let's take a look at that. Do you see the little white dots here up at the top? I didn't get a lot of growth. Usually I get more growth than this, but again, what's happening on this plate is we added antibiotic, so only those bacteria that took up the plasmid, that took up the pGLO plasmid, are going to be able to grow on this plate. The rest are going to die because of the antibiotic ampicillin. And so not every bacteria, as we can see, was transformed in that tube. Only some were. Okay, so we get some growth. Again, fewer than what I'm used to, but still. We got growth on this plate. We did not get growth on the minus pGLO plate, so it worked. Okay, remember this is our control here. If I got growth on this plate, something's wrong. The antibiotic's not working. Okay, so again, let me take my pen light here and I'll shine it. And again, I'll do this in the dark one more time. There is nothing glowing. These colonies are not glowing back at me. Okay, sometimes the camera makes it appear like they're glowing, but I promise you, they're not. Okay, all right, so let's look at my third set of plates here. So again, I've got plus pGLO, I've got minus pGLO. Both plates contain the same stuff. They contain L'Oreal broth, LB. They contain ampicillin, AMP. And what's added to these plates is the sugar arabinose, which shorthand we put ARA. That stands for arabinose. Okay? So let's hypothesize. What do we expect on these plates? Think about that. Should we have any growth on the minus pGLO plate? Hopefully you answered no, because the antibiotic ampicillin is present, and so this should be the same as that second plate that we just looked at. For the minus pGLO, they don't have the plasmid, so they don't have the ability to grow in the presence of ampicillin. Well, if they don't grow, they're not going to glow either, right? Nothing there to glow. How about the plus pGLO plate? Should you get growth? Yeah. Yeah, only those bacteria that were transformed and have that plasmid will have antibiotic resistance. So we should get some growth. Not as much as we got on just the LB plate. Okay, we're only going to get a fraction of that growth. Are we going to get glowing? Yes. Why? You have to be able to describe all this, right, in those questions that are coming up. So yes, you're going to get glowing because arabinose is present. So the sugar arabinose is necessary to turn on the green fluorescent protein gene. Okay, and once that gene is turned on, the green fluorescent protein is made. Now, with just normal ambient light, regular old fluorescent light in here, I'm not going to get any glowing. Do you see growth? Oop, where's my camera? Yeah, we got a couple, right? A couple little colonies. You can see they look white. They're about 1 millimeter in diameter. They're pretty teeny, maybe a 0.5 millimeter in diameter. Okay, they're small. They're there. They look just like the colonies from plate 2, which was just LB amp. Okay, what's the difference here? Well, let's shine our light. Yeah, okay. This worked beautifully. I'm not sure if you're going to be able to see that. I'll do it in the dark too. These guys are really glowing green. Okay. And again, I'm going to turn my light off in a minute, and I'll show you all the plates kind of together. So yeah, I'm getting glowing on here. So what's happening? The sugar arabinose is present, so the gene was turned on, the protein product was made. I also hit it with my ultraviolet light. So there's 2 environmental factors here that are required to get glowing. The sugar arabinose, which is in the plate, and the UV light. Okay, and then that green fluorescent protein will fluoresce green. Okay? Okay, let me pause this. I'm going to come back with the lights off and we'll do it again. Okay, so now I'm in the dark in the lab. And I'm hoping that you can see this. Again, the camera on the computer doesn't pick up great. I took some pictures of this and I think it's really evident in the pictures, so I'll post them on Blackboard as well for you, just so that you can see the difference. So this first one is going to be plus pGLO growing on LB amp ARA. And so these are the ones that glow. And let's see. Can you see them glowing? There's like 4 or 5 little dots up there at the top. There's one down here. Kind of hard to get it to, but again, these are glowing, okay? And then I'm going to go to my plus pGLO LB amp. This does not have arabinose on it. So the colonies are there. They're not glowing. No, nothing's glowing on here, okay? We do see some shimmer coming back off of the plate. Let's go back here. Again, let's compare them. See that? See that glowing? You can't see the green color really good, but see there at the top, there's 4 or 5 colonies. Okay, in person, they are fluorescent green. And again, on the plate without arabinose, they're there. I can see them better with the light on, but they're just a white color. Okay? So let me turn the lights back on and then we'll review. So again, just one more time for review. What we did here was a bacterial transformation. We got our bacteria to take up foreign genes, right, that were being carried on a plasmid. A plasmid is a small circular piece of DNA. It's a vector. It's a vehicle used to get genes into cells. Okay. The plasmid here was called the pGLO plasmid. It contains a couple of genes that we're really interested in that helped us in our work. The first gene was called the BLA gene, BLA gene, that codes for resistance to the antibiotic ampicillin. The second gene is the green fluorescent protein gene, and that requires the sugar arabinose to be present in order to be turned on. Okay. Once that green fluorescent protein is made, then it will glow only in the presence of UV light. Okay, so again, if I take my plate that's LB amp aura with the plus pGLO bacteria on them, under normal conditions, they're not glowing. It's only when I have the UV light present do I get glowing. Okay. What else did I want to tell you? So again, the minus pGLO tube, when I plated that on plates that had the antibiotic ampicillin present, I didn't get any growth. One of the questions that I'm going to ask you is, are there any plates that we didn't make that would have been maybe useful here? Okay, so think about it. We made just plain old LB plates. We made LB ampicillin plates. And then we made plates that were LB, amp, and aura. So they had ampicillin plus they had the sugar arabinose on them. Did I miss anything? Okay, so think about that. Was there anything else that I could have added in to look at anything else? Okay, so that's it. So you're going to go ahead and you're going to answer the questions activity. And again, I'm going to post some pictures of these up on Blackboard as well. So it'll just be a file there that you could take a peek. Again, the camera on the computer doesn't work fantastically. And so the camera on my phone is actually much better. So I'll show you those pictures. Okay, that's it.